The Hammond Lab, GEMM I riboswitches by Connor Hassinger, Effie Serrara, and Gabrielle Schatz. The lead researcher is Ming Hammond. They are based out of the University of California, Berkeley, and their two main research areas are RNA-based fluorescent biosensors and increasing sensitivity and selectivity of riboswitch biosensors. Ming also founded the website usestem.org, which is a free online resource for Bay Area K-12 students. And there's a picture of Ming. The GEMM I spinach. GEMM stands for Genes for the Environment, Membranes, and Motility. These are riboswitches derived from spinach, and this is Popeye and spinach. But wait, what is a riboswitch? RNA sequences that can be turned on or off based on the presence of small molecules. In some cases, fluorescence occurs. And this is fluorescence.
We're currently looking at a riboswitch called the GEMM 0970 spinach. It turns on when a CAG is present. The presence of a CAG shows that there is a fluorescent tag attached to it. A CAG is a molecule that's called a cyclic AMP GMP, and those are like small molecules. And it's a second messenger that regulates the vehicular in two ways: the cell chemotaxis and intestinal colonization. Also, there's extracellular electron transfer in a geobacter. We'll talk about what a geobacter is and why it is important. A geobacter is a bacteria that reduces metal. Geobacters use GEMM I riboswitches as receptors for a CAG. We talked about what a CAG is, and it regulates extracellular electron transfer, which we talked about in the previous slide. The way that they're applicable is when these bacteria can be used in toxic metal remediation to create electricity out of biowaste.
How are these biosensors made? So the lab made a library of GEMM I spinach variants by using the polymerase chain reaction, also known as PCR. The variants were then screened through fluorescence assays to determine which ones were the most selective for the CAG molecule. And the activation rates of the riboswitches were determined by measuring the fluorescence emission as the spinach variant was added to a solution of the CAG molecules. And then the deactivation rate was determined by measuring fluorescence as the CAG was removed from the solution by a buffer. And the most selective variant was then used in live cell imaging of E. coli. So here you can see that the riboswitch is off because it's not interacting with the CAG molecule. But when you add it in, it turns on the gene and you get a green fluorescent enzyme. The fluorescence of the riboswitches can be shown in figure 4b, where the live cell fluorescence of microscopy of E. coli is shown using two different variants of the riboswitch and two different enzymes. The first is DNCV, which is the CAG synthase. The second is WSPR, which is the CDIG synthase. And in parentheses, the intensity of fluorescence is shown, and it shows that the biosensors are more active when interacting with the DNCV molecule over the WSPR. In figure 4c, you can see the flow cytometry analysis of E. coli, and it's interacting with two different synthases. The modified DNCV synthase is very similar in fluorescence to the wild-type WSPR synthase, but is very shifted away from the wild-type DNCV synthase. And both of these tests show that the biosensors can differentiate between the two different enzymes producing CAG and CDIG, which are both very similar in structure. So what can these be used for? The new biosensors can be used in live cell imaging to determine the signaling pathways of CAG in bacteria, and the lab that developed it is also using the biosensors in their other research projects, like identifying new enzymes or other signaling mechanisms that regulate the amount of CAG in the cell. So thank you for watching our presentation on the GEMM I spinach riboswitches.